for everybody. It's my pleasure today to introduce Dr. Anthony Lozano. He is currently a postdoctoral researcher in the laboratory of Dr. Amaya Lujambio and Professor Joseph Lovett. Okay, is it correct? He is now working in the liver cancer program at the Icahn School of Medicine at Mount Sinai. Based on his CV, Dr. Lozano earned his bachelor in biology from the University of Perpignan. Sorry if my pronunciation of French is not so good.
Doing good.
Okay. And then he obtained also his master's in cancer biology and PhD in biology and health from the University of Montpellier in France. And then he works intensively in the intratumoral heterogeneity, RAS-MAPK signaling in liver cancer, especially in hepatocellular carcinoma. One of Dr. Lozano's current work focuses on how metabolic dysfunction associated with hepatitis, so MAS. Actually, this is also the topic of his presentations today. Reshapes the liver immune microenvironment. His work already presented in various high-impact studies, including in Life, Journal Hepatology, Gastroenterology, and Cancer Discovery. Today, Dr. Lozano will present his work entitled Impact of MAS on Tumor Immune Interaction during HCC Development. And then he's going to offer new insights how chronic metabolic liver disease shapes the immune landscape of liver cancer. So thank you for accepting our invitations. Can I just call you Anthony?
Yeah, of course.
Okay. Thank you for
thank you.
thank you that is dedicate your time this morning for giving this presentations to us. As I mentioned previously, it's going to be like 51-hour webinar. So you have like 45 minutes presentations, and the rest is going to be for the Q&As. So thank you. Time is yours.
Perfect. Thank you for having me. And as Cecilia mentioned, so indeed, this is like mostly my postdoc work that I did in the lab of Lujambio. And we're now trying to publish this data. So any comments are welcome. And so this work was mostly funded by Merck. And indeed, we studied the impact of MASH on tumor immune interaction during HCC development. So I guess most of you are familiar with metabolic dysfunction associated steatohepatitis, MASH, which formerly used to be called NASH. Just wanted to highlight here that this is a progressive disease, which obviously a healthy liver, but then people that have let's say bad diet habits and lack of physical activity will start accumulating in their liver. So this is called simple steatosis or MAFLD stage. But in some cases, people will progress to MASH, which is a more severe form of the disease, in which, in addition to steatosis, so they found inflammation, fibrosis, and in some cases, patient will progress to fibrosis and to hepatocellular carcinoma. But there are subset of patients that also progress from MASH to HCC right away. And obviously, there are factors associated with the progression of the disease, so genetic factors, environmental factors, and some complications like diabetes and obesity and cardiovascular diseases. And so in the lab, initially, so we were mostly interested in hepatocellular carcinoma. And so obviously, MASH-related HCC in that case. So just a few background of specifically MASH HCC. So it represents around 10% of the all HCC cases, but it is currently the fastest growing causes of HCC in Western countries. So as you can see here on the left, for example, in Europe or in America, so it's basically the incidence of MASH-related HCC, so the standard incidence rates between 2000 and 2021. So it's been increasing gradually in the last 2 decades. And so one specificity of the disease that unfortunately, so MASH HCC is usually diagnosed late in the course of the disease, and because of that, it has been associated with prognosis in patients. And, another specificity of that disease was the fact that it was recently reported in this paper in 2021 that typically MASH HCC were associated with poor response to anti-PD-1 is immunotherapy. And they are like at the moment, the current first-line therapy for advanced HCC is this combination of anti-PD-L1 and anti-VEGF. And there are more and more evidence that indeed HCCs are also poorly responsive to this combination, in which usually only 30% of patients respond, which highlights the need to have biomarker of response for these patients, but also novel therapies to overcome this resistance. Then if we have to discuss a little bit more in details about the interactions between the immune system and the tumor, so a few decades, 1 decade ago actually, in this review, people discuss how a tumor can actually escape from the immune system and how that could lead at some point to resistance to this immune checkpoint blockade that aim at restoring the capacity of the immune system to clear these tumors. Basically, so when you have tumor developing, so ultimately cancer cells will accumulate mutations, and these mutations will lead to the formation of what is called neoantigens, which are modified protein that are then presented at the surface of the cell by these MHC1 complexes. And so within the tumor cells will die. Tumor cells will die at some point, and they will release antigens, these neoantigens. And if this cell death is what is called like immunogenic, so encompassed with like some release of chemokines, so that this will attract antigen-presenting cells. These, which are typically dendritic cells or some macrophages, and they will phagocyte the dead cells and start presenting themselves to antigens at the surface of the cells. Might activate these cells that will migrate to the lymph nodes. And in the lymph nodes, if they encounter T cell-specific clones, they will prime these T cells and induce the activation in the lymph node. And then the T cells can travel back to the blood circulation, directly the lymphatic system, to the tumor. And then the activated T cells that will be specific a given antigen in which they were activated by will recognize the cancer cell and selectively kill this cancer cell. Example, one typical way to resist this T cell mediated killing is the expression of PD-L1 that will be the ligand for PD-1 expressed on T cells, and that will prevent T cell killing. So by blocking PD-1, the goal is to prevent this inhibition, and so release the brake on the T cells for them to kill the cancer cell. But obviously, for a tumor to develop, at some point, you will have to escape any of these mechanism here. But then, depending on the strategy that the tumor cell initially used to escape the immune system, immunotherapy might be inefficient. For example, if one of these steps are altered, you can release the brake by blocking PD-1, but the tumor cells won't have any T cell infiltrating anyway, so it might not lead to any response in the patient. Then, so the reason why I'm talking about all this, and that I mentioned earlier, MASH HCC. So overall, MASH HCC in the previous study have been described as an immunological disease. For example, there was this study by Ido Amit in 2021 published in Nature Medicine that showed that when hepatocytes accumulate fat, so hepatocytes are usually dying from toxicity, and this trigger immune infiltration. And notably, they showed that they found there's an activation of dendritic cells, which are these antigen-presenting cells that I just described earlier. And they suggested that this could lead to activation of T cells. And in this two other paper, it was found that in MASH, you had accumulation of what they call these autoaggressive CD8 T cells that were expressing PD-1 and CXCR6. And the reason why they call them autoaggressive is because they showed that they were actually able, in an antigen-dependent manner, so without necessitating the expression of antigen, to kill hepatocytes. So that's why they call this autoaggression, because these CD8 T cells, so through the release of acetate and extracellular ATP in the environment, they were able to kill hepatocytes, so autoaggress hepatocytes. And they showed in these two papers that these cells were contributing to liver damage. So, for example, when they both depleted dendritic cells or these autoaggressive T cells, they were able to reduce the progression of MASH. So they had like less severe MASH and overall less development of HCC at the end of the disease. And so indeed, this loop of inflammation and immune activation was promoting the development of MASH and MASH HCC. And in other studies, so it was demonstrated that MASH HCC were rather immune cold, and as I mentioned earlier, so resistance to immune checkpoint blockade. And so when we started the project, it was our project, so the question that we had here was like, when a tumor develops, so you have accumulation of antigens, and that should trigger an immune response overall that should protect against the development of liver cancer in that context. The question that we had was in this context of immune inflammation in MASH, how this antigen expression by tumors might affect this immune surveillance and this capacity of the immune system to respond to tumor development. And another question that we had was, which was also a big interest in the lab at the time, was how the genetic alteration of the tumor might impact this immune surveillance specifically. And so to address these questions, so we turn ourselves to experimental systems, so mostly mice model, which is big expertise in the lab. And so we wanted to have a model to generate MASH, obviously in animals. And so we took advantage of the Mount Sinai environment. And so we had this model, Mount Sinai, that was initially developed by the lab of Friedman, which used a Western diet that is high in sugar and cholesterol, and also drinking sugar in the drinking water, so fructose and glucose in the drinking water, and a low dose of CCl4, so carbon tetrachloride, which is a chemical toxic, so that didn't generate liver damage. And we did an injection once a week of this low dose to generate fibrosis as well. And so basically, here is just the histology of mice with typical normal diet, show diet, so typical histology of a healthy normal liver. And then on the right, you have the histology of a mice that were undergoing this protocol for 6 weeks, in which you can already appreciate the accumulation of lipid droplets in the liver, so extent of steatosis, and already some extent of fibrosis just after 6 weeks of this diet. And we wanted to make sure that we had also inflammation in this context. So I performed flow cytometry in these mice after 6 weeks of diet. And what you can appreciate here is that already after 6 weeks, we observed what was described in the literature. So an increase in dendritic cells, an increase in these autoaggressive XCR6 PD-1 positive CD8 T cells, but also an increase in Tregs, and these terminally exhausted CD8 T cells, so described by these markers here, suggesting already that you have enough chronic inflammation and immune exhaustion already after 6 weeks. And so we conclude that 6 weeks of this MASH diet already reproduced the immune landscape of MASH that was described in the literature. But then, so we wanted to be able to generate tumor in an efficient manner. also being able to manipulate the genetic alterations in these tumors and their antigen expression. So for that, so we had generated a model in the lab, which is based on isodynamic tail vein injections. If you're not familiar with this model, this relies on the tail vein injection of a large volume of mids DNA in the mice. So this injection will induce cardiac contraction, and all the volume that you inject will flow back into the liver, and inducing in vivo transfection of the hepatocyte. So this is a very convenient model to generate tumors in vivo. And basically, we use transposon-based vectors to overexpress oncogenes, for example, MYC or beta-catenin. And you can either combine two oncogenes like MYC and beta-catenin, or you can combine them with deletion of tumor suppressor gene using CRISPR-based vector. For example, we've been using deletion of p53, p10, KMT2B, KMT2C. So you can basically play with all the driver oncogenes or tumor suppressor genes that I found in hepatocellular carcinoma. So the lab has demonstrated that this model kind of like recapitulates the heterogeneity of HCC that are found in patients. And we were able to generate like subtype marine tumors that correspond to the human tumors. However, so when we were trying to study the interaction between the tumor and the immune system, one of the observation that we had was that so these tumors develop quite fast. And because of that, we found that they don't really express antigens. So you don't really have immune infiltration. So it's just two genetic hits usually that are not very immunogenic. And so these mice were not no immune infiltrate and were not responding to PD-1 or any immune checkpoint blockade. So the strategy that we then developed to overcome this limitation in the lab was to use this construct in which we fused the MYC oncogene with either a non-immunogenic luciferase, or luciferase just allowing the tracking of the tumor in vivo using this imaging, or this other version of the luciferase that we called LucoS, and that was initially developed in Tyler Jack's lab. And so this luciferase is fused to three model antigens, ovalbumin, Y, and synfecal. That these are like very strong and potent antigens that immunologists use a lot to mimic this more clearance in some case, or just like infections context. And basically, so the lab has shown in the previous publication that in some cases, for example, in this MYC-LucoS combined with p53, we had strong immune surveillance following the addition of the antigens. So the mice were clearing the tumor quite efficiently, but with different genetic models like MYC combined with beta-catenin, or MYC combined with the notch intracellular domain, there was immune escape. And so the expression of the antigens was not affecting the immune surveillance, and the mice still developed tumors despite this antigen expression. So, but this data that I'm showing you, so that was in the context of a healthy liver. So the mice were injected without any diet and previous liver damage. So the goal of my project was to understand what would happen in regard of this immune surveillance in the context of livers. And so this is what we did. So we did the either mice fed with a normal diet in gray, or this Western diet plus CCL4 protocol that I just described earlier. We perform hydrodynamic tail vein injection 6 weeks after the diet started, as I showed that we already had MASH-affected livers that were recapitulating the disease. And we started by testing three combinations that I just described before. So MYC combined with p53 deletion, beta-catenin expression or expression of the notch intracellular domain. And so here, without antigen expression, so what you can appreciate is that we didn't find much differences, maybe a slight tumor delay in the context of MASH with beta-catenin, but overall, survival of the mice were very similar. But then, so we added the antigen, and that was quite a surprise. First, what we observed is that all cases, despite the genetic alteration, addition of the antigen in MASH, which is here in green, lead to a very strong and efficient immune surveillance. So, to understand this data was quite surprising. So here, just to report this data. So this is the IVIS imaging of these mice, so fed with Western normal diet, 1 week and 3 week injections with both here Micluc beta-catenin or Micluc OS beta-catenin. So obviously wanted to make sure that the transfection efficiency was similar between the two conditions. This is just 1 week post-injection comparing the IVIS signal between here Micluc and Micluc OS. What's similar, but then you can see here, 3 weeks, the Western diet mice already cleared the tumors and had lost tumor signal. Indeed, so if you look at the liver of these mice, so the mice without antigen expression in both cases are indeed dying from the tumors all over the liver. But then in the Western diet condition, they were able to efficiently clear the tumors, and this aside from the fact that this were MASH affected livers, the livers look almost normal. We wanted to make sure that this was real and not a technical artifact, and indeed linked to this antigen expression. And so what we did here to deplete MHC class 1 through CRISPR against beta-2M, so which is part of this complex necessary for MHC class 1 and antigen presentation. So we combine a p53 CRISPR with a CRISPR against beta-2M with this Micluc OS, so enforcing the antigen expression in that case. And when we did that, we were able to completely restore tumor progression in this model, suggesting that indeed this is the expression of the antigen that is driving this tumor elimination here, which was like reassuring. Okay, we indeed, the phenotype we observe is linked to this antigen expression, might not be a technical artifact. But then still, when I first presented this data, so a major criticism what we had was that, okay, you are doing like a very short diet, so only 6 weeks of diet. You might have just like acute inflammation, and this is why you're getting rid of these tumors. What will happen if you do like a more chronic regimens and with a different diet eventually without the addition of CCL4, which might also create artifacts? And so we wanted to test then another different diet, which is a longer, more chronic diet that's been widely used also in the literature, which is this choline deficient diet. And we put the mice for 9 months under this diet this time. And as you can see here, so 9 months of this diet, here the mice are getting like completely obese. You have like mice that were more than 60 grams. And then again, we injected our plasmid either without antigen expression with this Micluc OS construct to induce antigen expression. And we were kind of like happy to observe that the data we observed was similar. Again, slight tumor delay with the Micluc beta-catenin for the mice undergoing this choline deficient high-fat diet. The mice that had antigen expression and undergoing the choline deficient high-fat diet, most of them completely cleared the tumors. So validating the previous result that we had with a different model this time. Then so we wanted to understand really what was going on in terms of immune infiltrate, why these mice were so efficient clearing the tumors. So we performed flow cytometry, 2 weeks injections, going back to this Western diet plus CCL4 diet. So first wanted to look at T cells response mostly. And so what big advantage of this system with these model antigens is that you can actually use tetramers to track track antigen-specific CD8 T cells. So T cells that will recognize specifically these antigens that we are inducing with the LucOS system. So here, the trimer against Fecal, Y antigen, overall that allows the detection of antigen-specific CD8 T cells. And what we could see is that already after 2 weeks, so we have a proportion of these antigen-specific CD8 T cells in the context of MASH. And looking a little bit at the functionality of these T cells, they were overall less exhausted in the context of Western diet fed mice by all these exhaustion markers here. And on the other hand, we had like more what is called like progenitor exhausted cells, which are cells known to respond quite well to PD-1 therapy and be able to trigger like efficient pool of vector-like CD8 T cells that then able to selectively kill the tumors. So this data was indeed going in direction to like increased immune surveillance and T cells vector functions in the context of this Western diet condition. But we wanted to validate the role of CD8 T cells functionally in this system. So for that, we used an antibody to deplete the T cells following the adrenaline telve injections. And so we compare this to their normal diet mice or just the Western diet plus the isotype control. And what we could see is that depleting the T cells in the system was indeed restoring the tumor progression at the same rate than the normal diet fed mice, suggesting indeed that this immune surveillance that we observed in MASH is promoted by the T cells in that context. And we did the same experiment, test the role of CD4 T cells, which can provide help with the T cell activation. But in that case, we found that this immune surveillance was independent of CD4 T cells, as the depletion didn't really affect the immune surveillance that we observed here. So indeed, these T cells require the CD8 T cells for this immune surveillance. But then we wanted to take a step back and see, okay, why these T cells are so much more efficient in this context of Western diet fed mice. And so we looked this time earlier in the course of the tumor development, so 1 week post adrenaline telve injection. And we looked at specifically antigen-presenting cells. And what we found, so here we were comparing the normal diet and Western diet look beta-catenin in that case, mice without antigen expression to the normal diet and Western diet LucOS, so with antigen expression. And what we found was an increase in type 1 dendritic cells, which are actually known to primarily activate CD8 T cells in the lymph nodes, specifically in that LucOS condition in the mice fed with Western diet. And this, looking a little bit at their functionality again, these cells, so we had a higher proportion of CR7 positive dendritic cells type 1, which are these activated migratory dendritic cells. And also there were higher amount of DCs expressing CD86, which is a known marker for activated dendritic cells. And so overall, increased activation and proportion of these DC1s in the Western diet condition. And we also validated this by staining, so just staining for CD11C, in which we found that we found the previous data with an increase these cells in the Western diet condition with antigen expression. Then again, we wanted to validate this data functionally by depleting these cells. So we use this time a genetic mice model, which has limitation, but is still a very useful tool to deplete cell type 1, so this knockout mouse. And again, we compare here in the microcos beta-catenin, so wild-type mice with pathway knockout mouse. And we found that in this pathway knockout, we were able to completely restore expression. So indeed, these dendritic cells are required also for this immunosurveillance, and they might be activating CD8 T cells in the lymph node of these mice. But then the question that we raised was, okay, how different are these dendritic cells in the context of MASH? And so for that, we got our hands on single-cell RNA-seq data that was comparing healthy normal diet mice with Western diet mice. In that case, they were tumor naive. And so here, what you have is just the clustering of the myeloid compartment, supervised clustering, and we could discriminate like different cell populations. And just looking at proportions, so we observed a trend for an increase in type 1 dendritic cells, which was significant in the context of 2 dendritic cells, but overall an increase in this, what is called MREC disease, which are these activated regulatory dendritic cells that were described by the lab of Miriam Merad. And so this MREC disease can arise either from type 1 dendritic cell or type 2 dendritic cells, and they are known to have of tumor immune responses overall, so suggesting that indeed we have a higher activation of oral disease in the context of a wet diet fed mice. But then looking a little bit more into detail, so we focused here on the type 1 dendritic cells, the pathway that were differentially regulated between the two conditions, so versus the healthy liver mice with the Western diet fed mice. Overall, we found way linked to inflammation, so interferon responses, TNF alpha, 6 JAK-STAT pathway. But then when we specifically looked at the differentially expressed genes, looking also a little bit at the literature, so 2 genes really got our attentions, which were this EL1B and LRP3 genes. So if you are a little bit familiar with immunology, so you might know that this is linked to activation of the inflammasome. And so when we went to the literature, we were aware of this paper from the lab of John Kagan, which have shown that the activation of the inflammasome in dendritic cells been associated with this, what they called an hyperactive dendritic cell state. And so this state relies on these 2 ligands, which are oxidized phospholipids, and the presence of LPS. Basically, when you have these 2 ligands, you have an activation of the inflammasome. So this NLRP3 that's just you before. And this NLRP3 activation induced the release of interleukin 1 beta. And they shown that this stimulates migration and the stimulation of long-lived T cell responses. So we were interested looking at the presence of these 2 ligands in the liver of mice. So we performed staining for LPS and these oxidized phospholipids. And indeed, we could observe that these 2 ligands were enriched in the liver of these mice. And so this is still a work in progress, but really functionally validating the role of these 2 ligands. So we're at the moment trying to prevent and block this oxidized phospholipid and see how it will affect this immune surveillance that we're observing in MASH. So this is work in progress. We are trying to functionally validate the role of these 2 ligands in this activation. But overall, so if I summarize what I've shown you so far. So we found that in the context of diet and mice fed with the diet, so we have this dendritic cell activation that might be linked to this increased LPS, which is a bacterial product, this oxidized phospholipid. And we found that this allows efficient activation of the T cells, so they're able to clear efficiently antigenic tumors. But then, so at the end, at the beginning, I showed you that MASH is a risk factor for HCC, right? And what we are showing here is that it seems to actually protect against HCC, which is a bit counterintuitive. And so when we to wrap our head around this, one of the hypotheses that we had was that this efficient immune surveillance might happen during HCC development at the very early phases. But then if you keep the damage going, what might happening is what is called immune editing, in which the cancer cell escape this immune pressure, will develop like specific characteristic that will allow them to escape the immune system. And that will generate tumors that are polyimmunogenic. And some of the characteristic of these polyimmunogenic tumors, notably the fact that they have DNA damage repair machinery that is proficient, so they're able to kind of like repair DNA efficiently to avoid accumulating too much mutation that will lead to this antigen expression and clearance by the immune system. And then they might have like low mutational burden because of that, and also lower neoantigen burden. So overall, allowing them to avoid this efficient immune clearance that might be taking place in the context of MASH. And so we wanted to validate this data and this hypothesis with the human data. So we looked in this cohort of patients across different etiologies at the proportion of patients that had mutations in these DNA damage repair genes. So when you have a mutation in any of these genes, so you will accumulate ultimately more mutations. And comparing the different etiology, we were quite surprised to observe that indeed, so literally no patients in this cohort that had mutation in this DNA damage repair machinery, suggesting that the tumors in the MASH context here quite efficient at repairing their DNA and then might accumulate less mutations. But then when we saw this data, we are curious to see, okay, what if we force one of these mutation in our mice model of MASH, what would happen? And so we decided to select this MLH1 mutation. So MLH1, if you're not familiar with this gene, so it's involved in this complex, the protein is involved in this complex of mismatch repair, so together with MSH2 and MSH3, so it's able to detect damage in the DNA and repair these mutations. And so when we combine this depletion of MLH1 with MYC, comparing normal diet and Western diet mice, we observe that reducing MLH1 mutation also delayed the tumor progression in MASH mice. And so that depleting CD8 T cells kind of like prevented this tumor delay. So this is the level that you have here in blue, so suggesting that this tumor delay is also linked to immune surveillance by CD8 T cells. And so we found also this to be an exciting experiment because it also showed that promoting DNA damage and mutagenesis and neoantigen in a more physiological manner rather than overexpressing strong antigens also generated this immune surveillance in the context of diet. But then overall, if you have less of this mutation, you will expect you will have a lower mutational burden. And so going back to this patient cohort, during the different etiologies, indeed we observed, even though there was not a very big differences in terms of median number of mutations, but the MASH HCC indeed had like lower mutational burden overall compared to alcohol and viral related HCCs. And in this other study where they were comparing totic liver disease, HCC, which is not pure MASH, so you might have also a mix of alcohol-related HCC here, but they observed that they had less predicted neoantigens overall in this hepatotic liver disease HCC. So again, going in direction that you might have like more immune surveillance during the onset of this development and clearing these tumor cells with high mutational burden and high neoantigens. All right. But then, so we found, okay, this is like quite interesting, but how can we use this information to treat eventually MASH HCC patients? And could we actually improve the response to immune checkpoint blockade by promoting antigen expression in MASH HCC patients? And so when we had this idea, more or less at the same time, there was a publication in Cancer Cell from Luis Diaz lab. It was two back-to-back paper, but I'm only showing one of them here. So this was not at all in HCC. So they looked at that in colorectal cancer, but they found this combination of drugs, which are actually chemotherapies, so that are well-known chemotherapies, but they haven't been that much combined before. So which is the cisplatin and temolizumab. And what they did and found that combining these two drugs, so this TMZ and CDP, as they call them, was inducing mostly a lot of frame shift mutations that are known to be very immunogenic because you completely change the sequence of the protein in that case. So you would expect that you generate a lot of neoantigen. And that was increasing the mutational burden of these cells like tenfold and inducing their like microsatellite stability. And they found in the papers, I'm not going to go into too much detail, but this combination was specifically inducing a loss of mSH2, which is also in that complex of DNA repair with one. So these cells are accumulating a lot of mutations. And in animal model, they showed that combining these two drugs with anti-PD-1 was improving the survival of the mice and their response to anti-PD-1. Or like, okay, in the context that we have in this MASH context where we have already active inflammation and immune surveillance, when we express antigens, it could be interesting to test these drugs and see if it would also improve response to anti-PD-1. And so just to show you, so in this model in which we don't have any antigen expression, so this make look 53, for example, even in the context of MASH, so the mice don't respond at all to anti-PD-1. Just looking at the flow data that we previously had, so this might be linked because they don't have any T-cell infiltration. So this is flow cytometry data just showing comparing like tumor naive mice with MASH, no tumor at all. And these look tumors, so they have exactly the same amount of T-cell infiltration as the tumor naive mice. And these T cells don't express PD-1, but then when you add antigen, so you have a huge T-cell infiltration in the cells, the T cells activated by these dendritic cells that updated the antigen, so they start expressing PD-1. So then we wanted to test this model in which you don't really have any immune response. How these two therapy combination might promote response to anti-PD-1 and immune clearance. So this is still very preliminary. So we still need to validate this data. So this is to take with a grain of salt. But what I can show you already is that here on the left, you have the IVs imaging and following the tumor progression. And it seems that this therapy was indeed very efficient at clearing and eliminating the tumors. So here, with the combination of platin and temolizumab plus anti-PD-1, so the signal is still going down. The mice are clear the tumor. Without anti-PD-1, it seems that it was also efficient, but then we had like some mice later on that rebounded and start developing tumors. So this is the survival on the right. Indeed, with the combination, we promote survival of the mice, but we still had like some animals that died from their tumors without adding the anti-PD-1 to promote this immune surveillance and immune checkpoint blockade. But we are currently validating this data, so we're going to do like some flow cytometry to really understand what's going on in terms of cell infiltration and compare to the normal diet-fed mice as well to see if these responses is indeed better in the context of MASH compared to unaltered liver damaged liver. All right, so just to summarize everything I showed you, so we found that in a MASH liver, we have this accumulation of APS and also oxidized phospholipid, and we are validating their implication in the activation of these dendritic cells. So when we induce tumor progression by adding antigens, so we observe this cell activation that promote antigen-specific CD8 T cells, and then are very efficient at clearing tumor in the context of antigen expression. But then what we think is happening is that in patients that will favor the occurrence of tumors, lower immunogenicity, so lower neoantigens, lower mutational burden, and overall that might trigger an immunosuppressive environment, so prevent T-cell infiltration because of that. And we think that promoting these antigens in one way or another could benefit to immune checkpoint blockade in that context. And we're also considering we have found that if we are able to validate the role of these two molecules in activating the ring cells, so these ring cell therapies, patients that don't have this overall active immune environment could be also interesting to test. Thank all the people that participated in this study. So initially people in Amaya's lab. Now I'm also working with Josep Llovet and Alice Kenforth's lab on that project, and all the funding and collaborators. And also thank you for your attention, and be happy to answer and address any question.